whatever you guys need to look at. So again, today we'll be talking about Mendelian genetics, but before that, let me just go over some announcements, just because emails wise, make sure you guys put which section time you guys are, because I have 3 sections on Wednesday and I have 74 of you guys, so narrowing it down as to which section you guys are will help me a whole lot, and it will help me get back to you guys easier. So just put in the timing. I know you guys are my 6 p.m. section, so please do that. If you guys haven't done quiz 1 and 2 already, please do it. It'll be closing out tonight. I'll have quiz 3 up. It'll be due next week because you guys have an exam coming up, right? So you don't want to be behind, huh?
Exam's Monday.
Oof. You guys feeling okay about that?
No.
I hope this session today will help with at least Mendelian genetics part. If you guys have any other questions from the previous sessions, please do let me know. Okay? Good. Okay. So that's pretty much my announcements for today. So I realized I made an error last week with this question. So which stage can cells either exit the cell cycle or become committed to completing the cell cycle? I accidentally said it was the S phase. That's where it can, so in the S phase, it can, so it's committed to cell division at that point, okay? So the answer to this question is actually G1, because it goes into cell cycle arrest, okay? Okay, great. I just wanted to make sure we were all on the same page. I was like, poof. I accidentally combined 2 different questions together. That's what happened here. Okay. So I had some, I just realized I never went over supercoiling and like linking number and all that fun stuff. So let me just go, we can go through like 2 problems of these guys. A circular bacterial DNA that is 2,000 base pairs in size has a linking number L of 100, 100. What does this tell you about the DNA molecule? Assume a Watson and Crick DNA structure of 10 base pairs per turn. So what do you guys think? It's okay if you don't have any clue. Okay, so linking number wise, linking number is essentially a term that kind of gives you the number of right-hand turns one of the strands of the DNA can make around the other, okay? So in this case, you have 100 right-handed turns of one DNA strand around the other. So that's what's there with linking number. Then you can tell the other component of twist because it tells you the number of base pairs and the number of turns it actually makes, right? Number of bases that are in per turn, right? So T would be the number of base pairs divided by number of base pairs per turn. So that gives you 200 turns in this case. Does that make sense so far? Okay. So then you have something called ripe. Ripe is actually taken as an expression of the number of supercoils, okay? So in this case, linking number equals twist plus ripe. To calculate out ripe, you would take linking number minus twist, okay? So in this case, it would be ripe equals 100 minus 200, which gives you negative 100. A ripe of negative 100 means that you have 100 negative supercoils. If this was positive, it would be 100 positive supercoils. Does that make sense? Yes? Okay. Sorry, I can't see all of you together, so, okay. So, okay, oh, keep forgetting to fix my animations. Anyways, so this is a similar question, except the difference here is the number of base pairs. You would get 100 turns per twist, 100 in linking number. And then in this case, right will be equals 0. So it's not really supercoiling. Does that make sense? Great, okay. Okay, so presentations. Let me just stop my screen share. So whoever wants to go first can go first.
All right, can you guys hear me well?
Yes.
All right. Hi, I'm Sophia. And let me share my screen so you guys can. Oh, it's not letting me share my screen.
Oh, whoops. Keep forgetting to do that. Okay. Now you should be able to.
Yeah, there we go. Okay. So let's start here. All right, so this is my presentation. So the question I got was, how many different kinds of gametes can be produced by an individual with the genotype homozygous dominant A, heterozygous B, homozygous dominant C, homozygous recessive D, heterozygous E, and heterozygous F. And then you have this list of choices. So the way that I went through to solve this particular problem is I worked with the equation 2 raised to the n, where n stands for the number of hybrid alleles that the organism has. So for this problem, we want to count how many heterozygotes it has. So we have a heterozygous B, E, and F, for a total of 3. So we do 2 raised to the n, or 2 raised to the third, which equals 8. So this organism can produce 8 different combinations of gametes, meaning our choice would be choice D. And there's that.
Good. Everyone on the same page with this question? Great. Okay. Who wants to go next?
I can go.
Okay.
Okay, give me a second. Okay. Oops. Okay, so for my presentation, I had the second question, and that was, if a typical somatic cell has 64 chromosomes, how many chromosomes are expected in each gamete of that organism? So I gave a little background and I said somatic cells are all cells except for germ cells. They contain 2 sets of chromosomes represented by 2n, where n represents the amount of chromosomes. Germ cells then undergo meiosis and form a gamete cell. During the process, the chromosome number is halved, and changing the ploidy level from diploid, which is represented by 2n, to a haploid, which is just n. And so therefore, if a somatic cell contains 64 chromosomes, the number of chromosomes in the gamete cell would be 32. So I just halved the number of the chromosomes in the somatic cell, and I got my gamete cell number.
Good. That is correct.
Okay.
Paige?
Yes, was I supposed to present today?
Yes.
Oh my gosh, I totally saw in my Blackboard that it was later in October.
What?
Yeah. If that's wrong though, I'm sorry and I wasn't prepared to present today.
Okay, at least have the PowerPoint up by later this week, okay?
Okay. I'm sorry.
It's okay. It happens with random assignments like this. Okay. What is happening? Just needed to double check something. Hold on. Okay, yes, I have you down for this week. Okay. Yeah, just upload the PowerPoint when you get a chance, okay?
Okay.
So what was that? Okay, so okay. Nope. Okay. Okay, so let's go into some Mendelian laws and have fun with Mendelian genetics. So what are the Mendelian laws of inheritance? Do you guys remember?
Are you talking about his like 3 postulates?
Yes, it's also called laws too, but yeah.
Okay, but the first one was unit factors exist in pairs.
Mm-hmm.
Do you want me to keep going? Or?
You can if you want.
All right, the second one is dominance and recessiveness.
Yep.
The third one is segregation.
Mm-hmm. Yep. So, okay, so I kind of put it out of order here, but law of segregation, it is essentially that alleles for each gene segregate separately, segregate from each other, that it ends up with each gamete having only one allele. Then the law of independent assortment involves genes of different traits separating out independently. Law of dominance, you see some alleles that are dominant while others are recessive. If an organism shows at least one dominant allele, it will display the effect of the dominant allele. And then, as we know, alleles come in pairs because you get one allele from mom, one allele from dad, and my phone keeps ringing. Sorry about that. Okay. So are we okay with the 4 postulates or laws? Okay. Okay, this does not. Technology does not like me today, guys. Come on. Okay. So I know you guys haven't really gotten into the product and sum rules in lecture. So I'm gonna go over this. So there are 2 ways to figure out the probability that you can predict genotypic and phenotypic outcomes. The first one is the product rule. In the product rule, it states that the probability of independent events both occurring together is the product of their individual probabilities. So for example, if you take 2 dice, the outcome on one of the dice is independent of the other, right? So in this case, if you probability of rolling a 4 is 1 out of 6 because it has 6 sides, one side that says 4. So probability of rolling 2 4s would be 1/6 times 1/6, which ends up equaling 1/36. Okay. So the next case would be the sum rule. The sum rule ends up stating that the probability of either of the two mutually exclusive events occurring is the sum of their individual probabilities. So probability of rolling 2 4s or 2 3s, that would be saying 1/36, because that's, you know, with that 2 4s, that's 1/36. Same goes with 3s because it's only one side each, right? So you would end up adding those together and get 1/18. So let's come to a question that is more genetics related, I guess. So in a family of with 2 children, what is the probability of there being 2 females? So what do you guys think? So this is our little Punnett square with different probabilities of getting females or males. Any ideas?
50% or one of the two.
It's kind of
50%. I don't have good Wi-Fi connection. I'm sorry.
That's okay. Okay, so let's
Is it
go.
1/4?
Yes, because you get half a chance of getting a female every time. So half times half is 1/4. With 2 females, it would be 1/4 that you would end up with that probability. That's the probability of getting 2 females. What about 2 females or 2 males? So we know that 2 females equal 1/4, right? So it would be,
half?
wait, say
sorry.
that again?
Is it 1/2?
Yes. So you would end up adding them together, right? So what about 1 female and 1 male? Any ideas?
1/4?
Close. Okay, so
Is
when
it
we
1/2?
have,
Sorry.
yes,
Is it a half? Okay.
actually. So with this kind of question, the order comes into play because you can either get a male first or a female first. So close. So essentially you get the same calculations with these guys, but then you have to add them up together to get the right probability. Does that make sense? So that's how it's 1/2. Okay. So what do you guys know about monohybrid crosses? We don't like monohybrid crosses, I guess. Okay. So monohybrid crosses, they're essentially trying to see what gives out, predicting the progeny of between individuals that have different alleles at one genetic locus interest. Okay, so essentially you're looking at one trait. You're crossing for one trait, okay? So in this case, this is like the color of the flower. We have 2 heterozygous flowers going on. So this would be a monohybrid cross. You get one homozygous dominant, two heterozygous, and then homozygous recessive. Okay, so this is the combination that comes out of two heterozygous. When you have either if you have dominant homozygous and recessive homozygous together, everything will just end up heterozygous. Does that make sense? Okay. So in this case, the probability of each of them, the progeny being homozygous dominant is 1/4, because you have half a chance to get a big P from each parent, right? That's the same thing with the homozygous recessive, because you get half a chance, right? So that gives you another 1/4. And then similar thing with the heterozygous. Again, in this case, order matters too, because like you can get either big P or the little p from the dad, or little p or big P from the mom, right? So, and so 1/4 plus 1/4, that gives you half a chance. So 2 going on there. So in this case, you have in phenotypic ratios, it's 3:1. Genotypic ratio would be 1 to 2:1. Okay, does that make sense? Are we okay? Okay. So now we have dihybrid crosses. Dihybrid crosses are essentially looking at 2 traits instead of 1. Okay, so mono versus di. Well, okay. So we go with 2 traits. Doing this cross is actually what allowed one to determine it shows us that law of independent assortment actually exists, because it shows that inheritance of one locus has no influence over the inheritance of another. And in this case, you would see a ratio of 9 to 3 to 3 to 1. This can be changed depending on other factors, but that's all you need to know at the moment, because you guys won't be getting to that until like next class or so. So don't worry about that. Just know that this is the ratio. It allows you to figure out law of independent assortment, okay? So we have this question. Mendel crossed peas having round yellow seeds with peas having wrinkled green seeds. All F1 plants had seeds that were round and yellow. Predict the results of test crossing these F1 plants. So what do we know about this question? Like, what's given already? And what do we know from looking at what the stuff is? Maybe writing this out might help a little bit. So we know that since all F1 plants are round and yellow, we know the genotypes for the parents are homozygous dominant and homozygous recessive, right? Because you have all of them being round and yellow. So you know that all your progeny is heterozygous. However, in some cases, you wouldn't realize what's going on. So we do something that is called a test cross. A test cross is to figure out what zygosity a phenotypically dominant individual has by breeding it with a recessive individual. Okay, so the results of this guy, if you're doing a test cross, that is what is happening. So if you have a heterozygous parent, you get half of these guys being round or wrinkled for each of them. And then with heterozygous again, you would get half either yellow or green. So then each of the probabilities would end up round and yellow. So half times half equals 1/4. Round and green, same thing. And then you would just continue on with that. Does that make sense? Okay. If you had a fruit fly or Drosophila melanogaster that was of phenotype A, so it's a dominant phenotype, what test would you make to determine if the fly's genotype was dominant homozygous or heterozygous? So how would we determine that?
Would you test it by heterozygous recessive?
Okay, so it's homozygous recessive, but yes. Yes. Okay. So essentially, if you have a fly that was AA, all progeny would be phenotypically A, because at the end of the day, if you have one dominant allele, it ends up masking the recessive, okay? And then if you had a heterozygous one, you would have half and half for each heterozygous or recessive. We cool? Okay. Which types of phenotypic ratios are likely to occur in crosses when dealing with two gene pairs for which all genotypic combinations are of equal viability? What do you guys think? What's the key word in this to answer the question right up the dot?
Would it be B?
So what, okay,
Is
not
it
B.
A?
Yes. Why is it A?
Because when you do the two dihybrid crosses, that's like the numbers that you
Yep.
get. Yeah, I don't know how to explain it better than that.
Okay, so the key term that we need to see here is two gene pairs, right? Two gene pairs means dihybrid cross. Dihybrid crosses have these this phenotypic ratio. Does that make sense? Okay, good. Just remember which one goes with which number of gene pairs. Okay, so I don't know if you go over this in lecture, but so with human blood types, that's one of the examples of dominance and recessiveness. So you have A, you can have either homozygous for A. Okay, so one of the things I should mention is that A and B are dominant and O is recessive. So in the cases of A and B, you can have a homozygous for A for both of them, or recessive, not recessive, heterozygous for A and O, okay? So this is just the way you would write for blood group, okay? So this is also with B, it's the same thing. You can have either homozygous for B or heterozygous. But then when AB comes together, they're both dominant. So they're going to show up together. Okay. And then O is homozygous recessive. Okay. So it's like sprinkled donuts here. So type A, you see all those sprinkles that look like the A antigen. B, type B has the antigens for B, or in this case, sprinkles. AB has both of them showing up, and then O has no sprinkles or antigens, okay? So we have a child that is AB and a mother who is AB. What are possible blood types for the father?
Could he be type A or type B?
And AB as well, AB as well, because they could get any of the alleles from the parents, right? What excludes them from being a father of this child?
O.
Yep. Good. So this is a question just written differently, but the same thing. In a disputed parentage case, the child is blood type AB, while the mother is blood type AB. What are the possible blood types for the father? Same thing, right? A, B, and AB. Okay. So I definitely know you guys haven't gotten this far, so don't freak out. I also have another video from YouTube on Blackboard, which I will make available to you guys later today. This is a way to calculate the probability of alternative ways to achieve that combination events. So binomial theorem is just, so instead of doing the product and sum rule like 1,000 times, it kind of goes with this to simplify it much down if it has two alternative outcomes. So in this case, you have, so A and B are the probabilities of two alternative outcomes. N equals the number of trials, okay? So don't freak out when I show you this next slide. Don't freak out, I promise. It'll get simpler. Okay, so with this, I promise you don't have to memorize all this. There's a trick to it. So with each of the terms, when you go through, it's essentially increasing by exponents, right? So for example, here, if you have the exponent to 3, you start off with the first term having 3, and then you go down with a, like 2, 8 to the 1, 8 to the 0 is 1, so you don't actually put a in there. So when you decrease an exponent, you increase 1 here, right? With the next term. And these are the coefficients that come with each term. Are we okay in how to kind of set this up, because it comes in handy when we do this problem? I promise I will have a better video for this, because I don't want you guys completely lost on me. So, okay, so here's a question that we can do together with this. What is the probability in a family of 5 children that 2 will be male and 3 will be female? So we use the binomial theorem in this case, because remember how we were talking about, so it can be different orders. With 5 children, that can be a lot of orders. So the binomial theorem kind of makes it less complicated in a way that we assign it. So A equals the probability of male, which is 1/2. B equals the probability of female, equals 1/2 again. Again, you can put it in any order as long as you make sure you have the right probabilities, numbers for each of the terms, okay? And then we're talking about a family of 5 children, right? So that's why our n value is 5. Then you go back, so then we're like, okay, so we need to find a plus b to the 5th power, right? So we go back to whatever that expanded version was. We write it down. And then we use these numbers, the 2 and the 3, will become our exponents for these guys. So that's how you would end up choosing for the probability. You would choose this term here because you want 2 males and 3 females. So in this case, you take those, you plug the probability values in, and then you multiply them all out, and that is how you get 10 out of 32 as your probability. Does that make sense? Or did I completely lost everyone? Are we okay? I promise I'll have another video up for this as well. Guys, say something.
I'm not really sure.
Yeah, I'm just gonna have to go through it a whole bunch more to like actually understand it.
Okay, yeah, I'll definitely put up another video. But essentially, this is the steps you would take. It's, again, this is only applicable if it's only 2 alternative outcomes. So in this case, it would be like a question where it would be like, what is the probability of having this many males and this many females? And then you would use this set here, depending on your n value, and then you just use that. Then you would just like, okay, so we know this is the numbers, and then you kind of just like narrow it down with that. Like your exponents would be these numbers right there. Are we okay?
Yeah.
Okay. Definitely let me know if you guys have any questions about this. I know this can be like complicated, and like if you hate statistics, you'll hate this question more than anything, but I wanted to make sure we actually did get to cover a problem in this, because I know Dr. Asuna did say in his concepts to know study guide, I wanted to make sure that you guys had at least one question in here. But yeah, I'll definitely put up another video from YouTube, which makes it 10 times more simpler than what I just did. Any questions so far, or do you guys just want to go through that on your own as well? Just like, okay. So if we had 5 children, and if we had 3 males and 3 females, well, no, hold on. No, not half and half, no, okay. What if we had 3 males and 2 females? Which term would we use in this guy?
The A cubed one.
Yep. So essentially that's how you would narrow it down, okay? It's like, just be careful of which exponents you're using and you should be okay.
Will they give you like the entire equation? Or will you have to know Pascal's triangle? You have to like write out Pascal's triangle.
You guys have to remember this is online, so you guys are open book. So don't freak out. This is open book considering it's online. So just make sure you know how to use it, and then you can just have it open somewhere.
Is this in the textbook somewhere? Or should I just like copy this all down?
I will have this PowerPoint up for you guys,
Okay.
so don't get freaked out. We cool? Because I have one more statistical analysis to go over with you guys. Don't hate me. I'm only helping you guys here. So what is the chi-square test? The chi-square test is intended to test how likely it is to observe that an observed distribution is due to chance. So in this case, so this is kind of our formula where O is the observed value, E is the expected value, so everything squared that over the expected value. So essentially in this case, it is allowing us to calculate the probability of a chance deviation of this magnitude from a 1 to 1 to 1 to 1 ratio, right? So you have the 4 different genotypes, and you want to see how they're going to end up showing all this, right? Like how much of a deviation there actually is with this, okay? Okay, so we have about, okay. So we have a sample of 200 with the observed values of these guys. What can be inferred? How much of a deviation is it from our expected values? So one of the things you can do is like look at the ratio of alleles. That kind of shows, okay, there's some deviation from a 1 to 1 ratio, right? So using this chi-square test, it ends up allowing you to see how much of a deviation it is from this expected ratio. So in this case, since we have 200 progeny, we would go with this ratio here because it's 200 divided by 4 out of this case, right? So your total progeny divided by 4, because we're having a 1 to 1 to 1 to 1 ratio, okay? So the next step would be applying this test to test the hypothesis that there is no significant difference between the measured values and expected values. So observed versus expected. Okay. So in this case, I won't make you guys do this out, but this is how you would calculate it out. Unless you guys have those fancy calculators that you can do them all at once. Just be careful with your numbers and signs because that will throw you off a cliff. But essentially, you calculate these guys out, comes in like this. So that's the first one for this specific genotype, right? Then it goes into like, so we calculate out the rest. And then you take the sum of all of these values. And then you get 9.88 for this guy, right? I know it's a lot of math in one go, but so that would be the chi-square value that you get, 9.88. So then to actually figure out if this actually works, okay, that's just our equation again. You use what is called a chi-square table. In the chi-square table, to look at anything, you have to do what is called degrees of freedom, or R value, which is essentially the number of genotype classes minus 1. So in this case, we have 4 genotypes. You take away 1, you have 3, right? So the degrees of freedom here, you come to it and then you kind of go through it, and we see where our the probability intersects with our chi-square value. So these are values that are related chi-square values. So in this case, when you look at 9.88, you go through 3, you're like, okay, yeah, no. But then you see 9.348. But then the next one above it is 11.34, right? So in this case, you would go with the one that is lower, but the closest never go above this number that you have. And when you look at that, you get this p-value, which is also meaning 2.5%. So typically the cutoff value, the standard is 5%, but it depends on the question too. So make sure you guys make sure that the cutoff is actually at 5%. If it's not saying anything, then assume it is. So in that case, so the hypothesis that there is no significant difference between these observed values and expected values end up rejected, meaning that there is a significance in the difference between these values. Does that make sense? No? Where am I losing you guys? We're all good? Oop, chat. Great, okay. So, okay, so considering I put you guys through a lot of crazy statistics today, as long as everyone puts in the word here in the chat, I am going to give you guys attendance points. So think I'm still missing like 2 people not writing up. One more. Who are we missing? Okay, I think everyone's in there. Okay. So do you guys have any questions, concerns? Did I freak you guys out? Any questions? But I know we don't meet again before your exam, so do you guys have any questions in anything that is genetics related? Whatever we've covered so far, I guess.
Elizabeth had a question in the chat.
Oh, whoops. It should be, unless Dr. Osuna says anything different in his lecture on Friday. It should be. There is also some parts with pedigrees that he might cover on Friday. I have my discussion video talking about different types of pedigrees. So make sure you guys are able to actually identify between them. I think he's gonna cover that on Friday's lecture, because that's towards the end of all of that. No, trust me, I would not do that to you guys. So the question was, does the quiz involve any of the math we did here? I promise no math on the quiz, just concepts, okay? I would not do that to you guys. Any other questions, concerns? Hating genetics? Okay, so that's all I have for you guys today. So if you guys have any questions, let me know. I'm pretty much here. Okay,
Thank you.
yep, not a problem.
Thank you.
Thank you.
Yep.
you.
Have a good day.
Thank you.
Yep.
Okay, so I just wanted to say that the confusion came because when I downloaded the Excel file to see like what question I was, it like auto went to the bottom. So I
Oh.
didn't see like the top, like literally the top 2 weeks I didn't see. And I honestly thought that I was on the 28th because someone's name is Gage. And I thought that
Oh,
that was a typo because like I couldn't tell that it was last name, first name.
oh, okay.
But it's still not an excuse to have like the incorrect date. So do you want it by Friday or by Sunday?
Friday would be preferable because I like to do my grading during the weekend.
Okay.
But if you can't make it in by Friday, just let me know.
I can have it to you by Friday, but it's gonna be like later Friday, like at night.
That's
'cause I
fine.
work during the day.
That's fine. I only grade on Saturdays. So,
And if you want me to like present still next week, the question, like it's up to you. I mean, I probably deserve to lose points over it, but if you want me to still present, I can do it next week.
I think we should be okay just because the topic kind of goes after the exam at that point. But definitely I would recommend doing a presentation too. Just because it is a requirement to actually speak it out. So,
Okay, so I can do it next week and just like make it quick because it's past like what we need to know.
yeah.
Okay. All right. Thank you.
Because we're still kind of covering Mendelian genetics questions next week. So it's like, eh.
All right. Well, thank you so much again. Like I didn't realize because I couldn't see it, but
It's okay.
all
We'll
right.
just
I'll
we'll
by
work with
okay.
Hmm?
I'll get to you by Friday.
it. Okay.
All right. Have a good night.
You too.